Drosophila is a species of fly that is generally known as the common fruit fly or vinegar fly. Experiments on this species often focus on the different pathways associated with determining its eye color. The wild-type fly has brick red eyes, and this color is controlled by two biochemical pathways: the omicron pathway, responsible for the synthesis of brown pigments, and the pteridine pathway, responsible for red pigment. For the purpose of this lab, we focused on the pteridine pathway since the omicron pathway produces pigments that are not soluble in water and therefore cannot be separated by paper chromatography. Mutations in the genes of the flies may result in non-functional enzymes, which result in colors that stray from the typical red-brown of the wild-type flies. If the mutation prevents the synthesis of a certain pigment, the eye color will be different. For example, if the brown pigment pathway is blocked, only the red pigment is made and the fly will have bright red eyes. If both pigment pathways are blocked, no pigment is made at all and the fly will have white eyes. In this experiment, we used 6 of each phenotype of Drosophila: isopropyl alcohol, fly nap, chromatography solvent, chromatography paper, aluminum foil, a beaker, cotton balls, dissection pins, forceps, white paper, pencil, staples, a UV lamp, and a stereoscope. To begin, aluminum foil was wrapped around the beaker to protect the chromatography chamber from light since the pigments are sensitive. Then a piece of paper was obtained and 6 marks were made 2 centimeters apart from each other, with the intervals being labeled with each of the phenotypes. After using fly nap to anesthetize the flies in their vials, 6 males of each phenotype were placed in the respective area of the chromatography paper. Under the stereoscope and utilizing the dissection pen sterilized with alcohol, their heads were smushed, releasing the pigments into the chromatography paper. After letting the paper dry for 5 minutes, it was rolled into a cylinder and stapled to keep its shape. Approximately 100 milliliters of chromatography solvent was added to the beaker, and the paper was placed into the beaker, allowing the liquid to cover the pigment. The top of the container was then covered with aluminum foil to protect the pigments. The solvent was allowed to run up the paper for about an hour, and after removal, the paper was allowed to dry overnight. Paper chromatography was used in this lab because it allows for the separation of different chemicals in the pigment. Each molecule travels at a different speed when pulled up the paper by the solvent. The lightest particles move more quickly and at a greater distance than the heavier particles, and thus all of the molecules make up the pigment are separated out. Due to the different eye colors, the flies showed certain pigments and not others. Wild-type eyes displayed almost all the pigments since no mutations are present. On the contrary, white flies have no pigment whatsoever, likely due to a mutation in a gene coding for the first enzyme in the pathway. Cinnabar eyes also showed no pigment, which can be concluded that they also have a mutation in one of the first enzymes in the pathway. Sepia eyes had no orange due to a mutation in the gene coding for the enzyme that resulted in the lack of drostophila. Even though in these results, vermilion and scarlet eyes lacked the violet-blue color, all the colors should have shown up, making the mutated gene likely to be on the omicron pathway. Sources of error could relate to inefficiency in smashing the eyes and the inexperience of the researchers when viewing the chromatography paper. Data could have been altered due to not understanding the full process and how to correctly find all the regions of the color on the paper.